Hi everyone, thanks for coming to my poster. My name is Helen. I'm the Senior Clinical Data Scientist here at n-Lorem. So in our N of 1 trials for most patients, this is the first time that their ASO has ever been given in humans. So that makes safety data essential. We need a system that reliably captures relevant data so we can quickly identify safety concerns that might require stopping dosing or see signals of benefit that guide whether we continue or escalate the dose. So today I'm going to present my poster, which shows how we built that framework for data capture and review. Now, there are some unique challenges when designing a data capture system for N of 1 trials. Each trial is individualized for the patient, so every protocol looks a little different, which means the data sets look different too. And that creates challenges for safety monitoring because it requires prioritization of different data across different patients. For example, in our patients getting ASOs administered to target the central nervous system, we may be looking at levels of cerebrospinal fluid, white blood cell counts, or protein, whereas for patients receiving subcutaneous doses of ASO that targets the liver, we may be looking at liver function tests. Now, to address this, we built a scalable end-to-end framework for capturing data in N of 1 trials. It's flexible enough to adapt to each patient's protocol, but standardized enough to capture some of the same type of data consistently across trials. The core of the system is a custom REDCap or research electronic data capture database for each patient, combined with remote monitoring and data cleaning, leading to automated data flows into PowerBI dashboards to allow for our quarterly data safety monitoring board reviews for oversight. And our goal is simple. We want to allow for accurate, timely, and intuitive safety review to guide decision-making. So step 1 is protocol development. Everything starts with a protocol. Each of our protocol designs is guided by our Study Treatment and Assessment Review, or STAR committee. And this committee includes both internal and external experts in clinical trial design and relevant outcome measures and assessments based on the ASO and that particular patient's phenotype or how their disease presents itself. Together with the principal investigator, or PI, the STAR committee makes sure that the protocol clearly defines efficacy and safety endpoints, when and how they'll be measured, and what data needs to be collected. And that foundation sets the stage for building the electronic database. And below we have a sample schedule of assessments from a protocol currently in use. Showing on the leftmost column is the list of relevant safety and efficacy assessments to be completed in the trial. And they include things like neurological exam, 12-lead EKG, CSF sampling for safety, among other safety measures. They also include assessments such as gait analysis or weighted communication scale, or WCS, to assess for changes in the patient's performance on things like walking and communication, among other efficacy measures. And this schedule shows when each assessment will be done and when each dose is, and that allows for careful monitoring of both safety and efficacy throughout the trial. Step 2 is building a data collection database. So once the protocol is finalized, we can then translate that protocol into a REDCap database. And we do this in 3 ways. First, it's customized. We build the electronic case report forms, or eSERVs, or data forms, that match the protocol. And this includes all assessments noted in the schedule of assessments that I showed in the last slide, and the assessment schedule, including dosing. We apply auto calculations and data validation rules customized to each particular assessment, such as requiring integer format for a particular assessment scores in order to reduce data entry errors. And second, this process is collaborative. When I design the database, I'll reach out to the site for input for assessments, their readouts, and their site-specific data units, such as those for local labs. Before the database goes live, we will train the site members on the REDCap database, and we do require that the PI reviews and signs off on the database as well before it goes live. And finally, collaboration on data also continues throughout the trial. The site reports to us all serious adverse events or serious medical issues within 24 hours of discovery. And the last C is consistent. So we ensure that the databases are consistent across patients by using things like standardized safety eSERVs across all patients, like vital signs, 12-lead EKGs, neurological exams, physical exams. But we also use standardized efficacy eSERVs as well for patients with similar phenotypes or similar disease attributes to allow for comparison. And finally, for consistency, we also create eServe completion guidelines for the site to ensure that when they enter data, it's entered uniformly across sites. This is a flowchart showing the workflow between us and the site during our trials. We design the database, review it with the site and PI, and then move it to production. The site enters data, and then we review and clean it, and then the data flows forward for analysis. And you can see how the responsibilities are shared. It's a back and forth process that ensures accuracy and timeliness. Now, I've previously discussed the database design in the previous slide, and I'll touch upon what happens after data is entered in the following slide. Step 3 is reviewing and cleaning the data. So once the database is live, then data monitoring can begin. And what this means is that we keep track of the data that's being entered, particularly safety data. Adverse events or AEs are any new health issues that a patient experiences during the trial. This can be related to dosing, such as back pain after a lumbar puncture for intrathecal ASO dosing, or something completely unrelated like the common cold. So we've set out REDCap so that the system automatically alerts us when the site enters new AEs or makes updates to pre-existing AE data. And that way our team can review and follow up quickly with the PI if needed. We also conduct routine reviews every quarter so we can prepare curated data sets for DSMB oversight. Finally, queries. These are generated manually when we find issues or are missing data. The sites will resolve them, and we track the progress of queries through an API-driven dashboard that updates daily. Here's an example view from a live REDCap database. Green pages mean the data is complete, and red indicates something is missing. This makes it very straightforward for us to spot gaps and follow up. You can see how monitoring and cleaning become visual, which helps streamline the process. Step 4 is exporting the data into dashboard. So when the database is cleaned, we move it into PowerBI, and we think about this step as the 3 Fs: feeds, format, and flags. So for feeds, clean databases feed into individual dashboards for each patient, and each patient's dashboard will have tabs for individualized safety data tables and graphs. Now for formats, dashboards include patient cards showing total doses, highest dose, number of AEs, and latest vital signs. The data is also structured into categories like labs, medications taken, and other safety events. Finally, flags. The dashboards will automatically highlight abnormal values and show trends like blood pressure over time or visualization of AEs by severity, which I'll show you in the next slide. And this allows us to detect potential safety concerns visually. And here's what a Power BI dashboard looks like for a patient in their AE tab. This patient only had one mild AE, an ear infection, which was not related to the study drug administration or study drug. The dashboard makes it easy to see the severity, timing, and whether it was considered drug-related. And this is the level of visibility we want for every patient, every trial. Step 5 is the n-Lorem Data Safety Monitoring Board, or DSMB. Ultimately, the cleaned and visualized data go to the DSMB, and this group is made up of independent experts, both internal and external to n-Lorem, and this committee has experience in ASO technology, trial design, drug development, and nano-rare diseases. Reviews are held quarterly using the Power BI dashboards that I just went through, alongside PowerPoint summaries that are made to include aggregate safety data as well as updates since the last meeting. And their feedback guides important decisions. For example, whether it's safe to continue dose escalation or even continue dosing after an AE. As of August 1st, 2025, we've implemented this process for 30 patients, and we expect more than 10 additional patients by the end of the year. Over time, we fine-tuned the process to make it more efficient and more reliable. And the key strength here is that it's standardized yet customizable and replicable. This means we can oversee safety in a timely manner, make clear data-driven decisions, and replicate the process across future N-of-1 trials. And this scalability is reflected in the graph. As the number of patients increases, the number of queries actually is going down. And that's partially due to sites getting better at data entry and partially due to us targeting queries more effectively. And in other words, the system is not only working, it's improving as we scale. We'd like to extend our sincere gratitude to the physicians and study teams at our participating sites for their dedication, hard work, and collaboration. But we're especially grateful to the patients and families for their trust, commitment, and participation. We could not do this without you guys. Thank you.